Welcome back to AllAboutRx. Today, we're tackling the crossroads of metabolism. We're going to cover how monosaccharides like galactose, mannose, and fructose feed into glycolysis, and then dive deep into the citric acid cycle. Let's break it down and master how our bodies turn raw cellular fuel into pure, usable energy in this explainer.
You know, it's wild to think about, but your body actually turns over its entire weight in ATP every single day. So what exactly is the final common oxidative pathway for carbohydrates, lipids, and proteins? How do all these different fuels power that ultimate energy engine? Well, to answer that, we're going to trace exactly how various sugars feed into the system and keep you moving.
Here is our roadmap for today. We'll start at the pyruvate crossroads. Check out the monosaccharide on-ramps, run through the citric acid cycle, tally up our ATP energy yield, and wrap things up with a high-yield quick recall. All right, stop number 1, the pyruvate crossroads. This is essentially the major metabolic branch point. Think of pyruvate as the ultimate metabolic traffic cop. It sits right in the center of the intersection, and depending on what specific tissue we're talking about or what state your body is currently in, it directs traffic in several totally different directions. Let's look at the options. Say you're sprinting and oxygen is low, boom, anaerobic conditions. Pyruvate turns into lactate. Need amino acids? Transamination turns it into alanine. But if you've got plenty of oxygen, it heads straight into the mitochondria as Acetyl-CoA for aerobic oxidation to power the TCA cycle. It can even form oxaloacetate, L-malate, or phosphoenolpyruvate to support gluconeogenesis. The huge takeaway here is that pyruvate adapts instantly to exactly what your cells demand. Moving right along to section 2, monosaccharide on-ramps, let's explore galactose, mannose, and fructose. We all know glucose, but how do these other sugars get onto the metabolic highway? Take D-galactose. It can't just merge directly into glycolysis. First, it gets phosphorylated at carbon 1 by galactokinase. Then it takes a multi-step detour using the GALT enzyme and a specific epimerase, until finally it's converted into glucose 6-phosphate. At that point, it safely joins the main glycolytic pathway. And here is a massive clinical pearl for you. What happens if there's a roadblock in that pathway? Well, a defect in galactokinase, GALT, or the epimerase causes a condition called Galactosemia. This is serious. It leads to a really dangerous buildup of galactose in both the blood and urine. Okay, what about mannose and fructose? They've got their own specific on-ramps to become glycolytic intermediates. Mannose keeps it pretty straightforward. It's phosphorylated by hexokinase and then isomerized right into fructose 6-phosphate. But fructose routing is highly tissue-dependent. Check out this crucial distinction. In your muscles and kidneys, hexokinase just grabs the fructose, makes F6P, and tosses it straight into glycolysis. Super easy. But the liver uses a completely specialized fructokinase pathway to make fructose 1P. Then aldolase has to come in and split it, and you actually need triphosphokinase just to finally join glycolysis as glyceraldehyde 3P. The end goal is exactly the same, but the biochemical paths are beautifully adapted to different organs. Next up, section 3, we've hit the main highway, the citric acid cycle, our final common pathway. All those convergent on-ramps we just talked about, they all lead right here to the mitochondrial powerhouse. Often called the Krebs or TCA cycle, this elegant 8-step loop takes acetyl-CoA and systematically converts it into carbon dioxide and water. This is the final oxidative pathway. Carbs, lipids, proteins, they all eventually funnel in right here. Now, while every single step in this cycle matters, I've got a high-yield tip for you. Commit these 2 specific enzymes to memory. First, succinyl-CoA synthetase. Why? Because it is literally the only substrate-level phosphorylation step in the whole cycle, and it kicks out a molecule of GTP. Second is succinate dehydrogenase. This one is unique because it's the only FAD-linked step, and get this, it's embedded directly in the inner mitochondrial membrane. It acts as complex 2, linking the cycle straight to the electron transport chain. Moving to section 4, ATP energy yield. Let's count up those energy molecules. After doing all that biochemical heavy lifting, what's the ultimate payoff? The magic number is 12. You net exactly 12 ATP for every single turn of an acetyl-CoA molecule through the cycle. Let's break the math down like a ledger.
In one single turn, the cycle produces 3 NADH molecules, which cashes out to 9 ATP. You get 1 FADH2, giving you 2 ATP. And remember that 1 GTP we just talked about? That provides exactly 1 ATP. Add the whole shebang together, and boom, 12 ATP per turn. It's just an incredibly efficient engine.
Okay, bringing it home with Section 5, quick recall. Let's lock in these high-yield summaries for maximum retention. Now is the time to take a screenshot. This is your ultimate study guide for everything we've covered today. It visually maps out pyruvate's fates, those distinct monosaccharide pathways, and the absolute highlights of the TCA cycle. Keep this handy for your final review. Let's rapidly hit those high notes one last time to cement them. Pyruvate is your massive metabolic branch point. Remember our clinical pearl. Defects in the galactose pathway cause galactosemia. Muscle and liver use distinctly different pathways to process fructose. The TCA cycle is your final common oxidative pathway for carbs, lipids, and proteins. One single turn nets you exactly 12 ATP. And do not forget, Acetyl-CoA synthetase is the only substrate-level phosphorylation step. Repetition is absolutely key here, so keep reviewing how these substrates converge and precisely how much energy they yield.
So, that brings our biochemical roadmap to a close for today. But it leaves us with a pretty fascinating question to ponder. How might these intricate metabolic pathways adapt during prolonged fasting or, say, extreme physical exercise? Just imagine how these incredibly efficient enzymes shift gears when pushed to their absolute limits. Anyway, what part of the TCA cycle do you find most confusing? Let me know in the comments below. Don't forget to subscribe to AllAboutRx for more high-yield medical content. See you on the next explainer.